A good morning or afternoon to you. Thank you for joining us. I'm Jan Reichert, Director of Business Intelligence at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics relating to antibody discovery and development. Our expert speakers, Vincent Pai and Dr. Liang, will offer advice on how to accelerate therapeutic antibody discovery using innovative technologies to transform hit generation and lead characterization. Please note the webinar is being recorded. Please do add any and all questions to the Q&A box in the viewer, and those questions will be answered at the end of the webinar. Without further ado, I'll now turn the show over to our speakers, starting with Vincent Pai.
All right, thank you, Jan, for that introduction and the opportunity to speak today. My name is Vincent Pai, and I'm a Director of Product Management at Bruker Cellular Analysis. I'm grateful to have the opportunity today to present on Bruker's behalf and to speak with one of our incredible customers pioneering new innovations into antibody discovery. Before we hear from Dr. Liang's work, I wanted to take several minutes to quickly explain the key technology fueling rapid antibody discovery breakthroughs in my presentation titled Optofluidic Discovery, Antibody Discovery on the Beacon Platform. Wanted to start by first talking a little bit about who we are. So just over a year ago today, actually, two single-cell functional characterization platform companies, formerly known as Berkeley Lights and Isoplexis, were acquired to form a new subdivision within Bruker called Bruker Cellular Analysis, and I'll be calling it BCA for short. BCA is Bruker's first strategic entry into the single-cell multi-omic space. And we're really excited to be part of Bruker's motto of continued innovation with integrity, to continue developing new innovations to further push single-cell characterization applications. As we did in the past, we'll continue to provide scientific leaders and innovative researchers access to single-cell functional multi-omic platforms, both the IsoSpark line of systems and products for bulk and single-cell proteomics, and the single-cell optofluidic Beacon platform, and products for antibody discovery, cell line development, and T-cell profiling. These two platforms are at the forefront of Bruker's strategic entry into that single-cell space, and both the technologies will continue to provide those innovations across a wide range of life science applications, even beyond those listed here. Today, I'll be focusing primarily on the Beacon optofluidic platform and our antibody discovery application, which enable rapid, species-agnostic, function-first, single B-cell screening, powering monoclonal antibody discovery primarily for therapeutic, diagnostic, and reagent uses. And so some of the key things that the Beacon technology offers in terms of its functionality include large comprehensive assays, broad species compatibility, very high throughput screening for single cells, automated sequence recovery, and a rapid single-day workflow. And taken together, this allows us to deliver to our customers using the system to be able to prioritize functionality first so that they can focus on screening antibodies based on their function with back-to-back or multiple sequential assays to gain deeper and more relevant antibody insights. It allows them to maximize discovery success for every campaign, achieving exceptional results and uncovering diverse antibody hits, even with the most challenging targets, and allowing you to screen through multiple different species and targets. It enables users to expand assay applicability by empowering screening with small volume samples and customizable assays suitable for various therapeutic antibodies. And of course, this enables fast and an automated workflow that employs a single-day, highly customizable workflow with automated sequence recovery, significantly reducing both time and costs in the entirety of the workflow. And so these taken together enable the OptoB discovery product portfolio to enable researchers to rapidly compress traditional timelines from an immunized sample down to sequences of lead molecules, down from months to just weeks, while dramatically increasing the opportunity to screen more deeply for these functional antibodies. At a high level, this is kind of what the OptiB discovery workflow looks like, starting with an immunized sample on the left. VCA then provides reagents and know-how to help isolate, if needed, and activate antibody-secreting B cells from those samples to be loaded onto the Beacon system. Once those B cells are positioned with a single cell in a single nanopen, assays are performed to assess a variety of phenotypic and other functional characteristics of secreted antibodies, which all layer together and inform hit selection. Hits can then be recovered into well plates using a number of different methods, and then further downstream molecular biology processing is supported by our products and reagents to prepare libraries for sequencing. Depending on the sample and the sequencing, this whole process can take under a single week, especially with the on-Beacon workflow and screening taking as little as a single day. Zooming a little bit into that first step of this process, B cells are typically isolated from a variety of different immunized samples or even convalescent patients. And we primarily derive either memory B cells or directly antibody secreting plasma cells from bone marrow, splenocytes, or PBMCs. While our protocols and validation data typically showcase memory B cells and plasma cells that haven't gone through pre-enrichment for antigen specificity, these methods can be employed to further increase the likelihood of finding ideal hits and sequences. Once we get to antibody secreting plasma cells or B cells, we can then load those into our optofluidic chips, or these OptiSelect chips on the optofluidic system. And so then diving a little bit further into the system itself, so we have a really large instrument and inside that instrument, we can run up to 4 optofluidic chips that are situated inside to take advantage of the instrument's optics, liquid handling, and software. We can run up to 4 of those chips in parallel in our flagship model, with software managing the automation necessary for screening at the high throughputs and complexity that we offer. Within the optofluidic chips, there are 2 key innovations that deliver the single cell functional biology and applications that we offer. And so the first on the left is the horizontal nanopen and channel design, which exploits tiny volumes and laminar flow of microfluidics to enable real-time measurement of secreted molecules, cell-cell interactions, and diffusion-led cultures for even up to 28 days. The second innovation is on the right with our optoelectropositioning, or for short, OEP technology, which enables manipulation of single cells or beads with patterned light, enabling the finest control in creating single cell experiment conditions at massively parallel scale. And so you can kind of see here that we're able to load those cells into the main channel and then use light to pen these cells. And so specifically for antibody discovery, we have 2 flavors of chips that we primarily use, both the OptiSelect 11K chip and the 20K chip, which of course offer 11,000 pens or 20,000 nanopen chambers, respectively. And what we do is we leverage the OEP to load these plasma B cells that are imported into our system at massively parallel scale. And users often see about 50 to 75% penning efficiencies, resulting in about up to 60,000 single B cells when being screened across 4 20K chips. And so once these cells are loaded into these nanopen chambers, we can really start to create and assess the antibodies that are being secreted. And we start with the simple binding assays that really assess a key antibody characteristic, which is antigen specificity. By coating streptavidin-coated beads with biotinylated antigens of interest, a simple sandwich assay can be performed in the channels of the chip that leverages the active secretion of the B cells, the small volumes that yield high concentrations of secreted antibodies, and the diffusion of those secreted antibodies out of the nanopens. By importing these beads with that fluorescent anti-IgG antibody, nanopens containing antigen-specific clones can be identified by these what we call fluorescent blooms above these nanopens, and both manually by the user or leveraging our sophisticated machine learning model, you can automatically detect those blooms. These assays can be performed in as little as 15 to 30 minutes using just 30 to 50 microliters of reagents per chip, enabling rapid screening across every B cell that's loaded on those chips. Then, if you want to screen for another molecule, you can simply just flush out those beads and reagents and import new beads that are coded with a different antigen, as well as new secondary antibodies. And so you can run another sequential back-to-back assay to screen those same B cells for yet another target. Taking binding assays one step further, we can take another straightforward approach where we use the soluble where the soluble antigens are actually are fluorescently labeled, and we use anti-IgG capture beads as well as anti-IgG fluorescent secondaries in different colors to enable multiplexing here. And so this allows us to not only be able to detect cross-reactive antibodies, let's say against mouse, cyno, or human antigen targets, or different mutants and similar molecules, we can actually start to assess the antibody affinity for some of these soluble antigens, allowing users to get a quick and early measurement for the strongest and even the weakest binders within a single sample. So I'm going to dive a little bit deeper into that antibody affinity. This is one of the newest capabilities that we released recently. And so just another schematic of our assay here, but really trimmed down to just a single antigen, again, directly fluorophore labeled, and with these anti-IgG secondary. And so to perform an affinity assay measurement, we leverage this specific design. And to note, the biotinylated antigen-coded beads doesn't work for this particular measurement. And what we're able to do is we perform a more sophisticated imaging process or image process analysis where we are measuring the brightness over time and over distance for each of these blooms. And so when we plot the brightness in the y-axis versus the distance of that brightness from the bloom source, and we plot that at different time steps accordingly, what we can actually start to do is fit this against a standard diffusion model, which yields the scores for both the IgG bloom assay as well as the antigen-specific bloom assay. And taking those together, what we're able to do is if we normalize those antigen bloom scores, seen in pink on the left here, by the IgG bloom scores, seen in green on the right here, then essentially what we can calculate is a percent of IgG bound to a particular antigen at any given time in our assay, which we call the beacon affinity score. This beacon affinity score is directly proportional to the KD of the antibody-antigen interaction, which is also an equilibrium measure of the percent of or the amount of bound antigen for any given concentration of antibody. And as such, higher beacon affinity scores are likelier to have stronger binding affinities. Okay, so I'll go really quickly through some of these here, and I apologize again for that technical difficulty. Yeah, so jumping back to that OEP and what that actually looks like. So we can import these plasma B cells or these antibody secreting B cells into the main channel, and we can leverage our OEP to load up to 60,000 of these single B cells across our 4 OptoSelect 20K chips in massively parallel scale. Yeah, so on the left here is kind of a schematic of one of the simpler assay designs that we offer, where you have a biotinylated antigen that coats the streptavidin-coated beads, and when you import these beads into the main channel with an anti-IgG secondary antibody, then secreted antibodies that are antigen-specific will actually start to accumulate on these antigen-presenting beads, and accumulate the signal from that secondary antibody, and you'll create these blooms above these nanopin chambers in as little as 15 to 30 minutes, using only 30 to 50 microliters of acid reagents at a time. And so in rapid succession, what we're able to do is then assess for antigen specificity in your first assay. And if you would like to screen for a different antigen or a different target, you can simply flush these beads and reagents out of the main channel and import new ones and start imaging for that second assay against that second target to perform those serial back-to-back assays. Hopefully that changes. I think it's stuck again. All right, great. And so then the second, taking this one step further, another straightforward approach for us to assess for antigen specificity against soluble antigens is to leverage an anti-IgG capture bead and import fluorescently labeled antigens or secondary antibodies with each molecule type in different fluorescent colors. This enables multiplexable assays that can quickly determine cross-reactivity against different variants. And so you can think about your mouse, human, and cyno versions of your antigen, or maybe you have different mutants or different or highly related molecules. And in addition, this particular assay type enables the measurement of relative binding strength or affinity of the secreted antibodies, allowing users to get a quick and early measurement again for the strongest and the weakest binders against your particular antigen. And so, okay, hopefully this will change without me restarting. Okay, wonderful. Okay, so diving a little bit deeper into that specific affinity assay, and just simplifying that schematic to just have a single antigen, directly fluorophore labeled antigen in pink, and an anti-IgG secondary in green. What we're able to do is measure the brightness over time and across distance from each of the nanopen chambers individually. And so by plotting that brightness over distance, so that's y-axis in brightness, x-axis for the distance across, from the nanopen, and then over time we plot each of those brightnesses as that bloom gets brighter, as the bloom gets bigger. What we are able to do is perform a fit against the standard diffusion model to yield scores for both the IgG assay as well as the antigen assay for every nanopen chamber that's screened. And then by calculating, once we've calculated those IgG and antigen bloom scores for every nanopen chamber, we can normalize the antigen bloom scores in pink over here by the IgG bloom scores for the respective pens in green here to yield a beacon affinity score. And so you can kind of see that here. And what this beacon affinity score is essentially a percent of IgG secreted that's bound to the antigen at any given time in our assay. And this measurement, or this beacon affinity score, is directly proportional to the KD of the antibody antigen interaction, which is also a measure of equilibrium, our equilibrium measure of the bound antigen for any given concentration of antibody. And as such, we can expect that higher beacon affinity scores are likelier to have stronger binding affinities. And so we validated these beacon affinity scores against industry standard tools. Shown here is that comparison against Octet, which uses biolayer interferometry. What's not shown is one of these, the plot in the middle for the antigen TIM3 that was measured against SPR, or surface plasmon resonance. And we did this across 6 total different antigens, just 3 shown here. These error bars represent the blooms, or the bloom scores, the beacon affinity scores from different pens that ultimately yield the same exact heavy chain and light chain sequences, so that we could assess the variability of the beacon affinity score against the actual KD measurement via BLI. And while this data certainly isn't as accurate nor as strong of a correlation as other industry standard systems, we're still really excited by this measurement because this can be generated within minutes, and in particular, weeks to months in advance of many antibody characterization workflows, and particularly at a scale and throughput that's unparalleled by any other technology on the market. So for the 60,000 B cells that we're able to load, we yield measurements for all of those in that single day workflow on the Beacon system. And so then taking this one step further, we've talked a lot about recombinant antigens or soluble antigens, but we can also assess, we're not limited to that, and we can extend this to membrane-bound targets as well, especially for GPCRs, transport proteins, ion channels, and as well other transmembrane proteins. Here, endogenously expressing, overexpressing cell lines with high amounts of surface antigen can be used instead of those antigen-coded beads to identify antigen-specific clones. And of course, these assays can be layered on top of those soluble antigens simply by importing those beads later and flushing these cells out. And finally, we do offer the ability and have demonstrated the ability to do even more complex assays. That said, these do require quite a bit more optimization and development for each particular for these particular targets. So these are very advanced, but these do allow us to assess really functional characteristics, including blocking, reporter gene or for gene assays for agonistic antibodies or antagonistic antibodies, and even antibody internalization more recently. I'm going to jump really quickly into how it is that we recover then these B cells. So after performing single or multiple serial function first assays, we can recover these BCR sequences leveraging fully automated and user-defined processes. And so the simplest method just largely leverages the OEP technology to move the cells back into the main channel. And then we distribute those single cells into single wells to maintain heavy chain, light chain monoclonality. Of course, to overcome some of the viability concerns with single cell unload methods, we can leverage our OptoSeq BCR technology to perform reverse transcription on chip to capture mRNA and then turn that into stable cDNA to recover those beads and those sequences later. Of course, we provide know-how and reagents to help support single B cell sequence recovery from a well plate, which can be challenging, and we primarily target these for and amplify these BCRs for Sanger sequencing. Of course, taken together, we really pride ourselves with being able to generate quite a bit of functional assay data for every BCR sequence that we recover. And when we combine these, we can start to create very rich data sets that enable us to take and gain new insights into antibodies that we should take forward into development, as shown with one of our partners, Pipe Bio here. And of course, I wanted to cover this final slide here about our technical prowess here. Is that we believe that the scale of the combined functional and genomic data is critical to address the major bottleneck in antibody development moving into lead optimization. And so with advances in AI and machine learning, of course, these data-hungry models not only require BCR sequences known to a single binding to a single antigen, but of course, deeper functional characterization for every sequence that informs how well the antibody actually binds, what epitopes they bind to, and what functions they serve. And so clustering only on those sequences based on binary binding, not binding information, only gets you so far, despite being able to see certain clusters of information here. The traditional process of then hand selecting just a couple different antibodies that potentially represent each of these families for characterization still is very limiting. And so this cherry picking process isn't going to really carry us through into the future. And so one of the things that we pride ourselves in is the amount of data that we generate, that functional data that we generate, that's linked to each of the antibody sequences that we generate. And therefore, this allows us to layer in yet another dimension or several dimensions of functional characterization for each of these sequences that allows us to really dial in to certain clusters and really make informed decisions and novel insights into leads that we want to carry forward. And so moving towards the close of my session here, we do provide quite a bit of different products to enable both end-to-end solutions, both for mouse, human, and rabbit antibody discovery. But this modular approach also allows you to mix and match as you need to enable new species or new assays for different targets and new ways to recover BCR sequences that work best for your work stream. And of course, we're really excited to hear from Dr. Liang in a couple minutes about how his team at GenScript ProBio can leverage this modularity and this customizability to offer even greater advancements within the antibody development process to their clients. Finally, I wanted to close my section by just showing a timeline here of some of the major product extensions and features that our team have been able to release since we first released our first capability in 2018. And so, of course, true to Bruker's motto of innovation with integrity, Bruker Cellular Analysis has been and will continue to continuously innovate on the Beacon platform and continue to deliver new value to its users, especially as we build towards a total solution for any antibody discovery need. And with that, I wanted to pass this over to Dr. Liang. And I apologize for some of the technical difficulties that we encountered.
Sure. Hi, everybody. Thanks, Vincent, for a great talk. Let me show you my screen. So by the way, the name of my company is called like ProBio. So that's why, everything, we trademarked platforms or, capabilities is kind of initiated with like a pro. Yeah. So today the platform I'm going to talk to you about is like a ProSpeed single B cell screening service, which is based on the Beacon, but it's an upgraded protocol based on the Beacon system for antibody discovery. Yeah. A few words about who we are. So basically, as Vincent mentioned, we are like a subsidiary company of GeneScript, and for the Biologics Discovery Center I'm in charge of. We are like a team of like over 120 scientists and with a history of over 20 years. So in 2004, we started from the hybridoma platform, and then gradually we established other platforms for antibody discovery, including phage display and also the single domain antibody discovery. And in 2019, we introduced the Beacon single B platform, starting to offer the single B cell based antibody discovery. And recently, we extended our service to some other advanced modalities, including the CAR lead discovery and also the one-stop ADC lead discovery. But basically, we kind of support all kinds of antibody discovery related services, including lead generation and antibody lead optimization, humanization, affinity maturation, and also in vitro and in vivo functional characterization of antibody leads. So we offer a tailored discovery strategy for novel biological modalities. For example, for the monoclonal antibody discovery, now we developed mRNA and VLP-based immunization strategies to improve the positive rate of hits. Yeah. For cell therapy, we offer like all-inclusive, CAR T or CAR NK bioassay services to help you, discover CAR lead, from like lentiviral construction to like T cell transduction and functional assays. And we also support bispecific antibodies functional characterization, and recently antibody drug conjugates lead discovery. Yeah, looks like I cannot do the full screen mode. Yeah. So now I'm going to briefly talk about an upgraded protocol called ProSpeed to address some of the challenges in the single bead technologies, or based on the version of Beacon we are using it currently. As we know, most of the antibodies approved by FDA is discovered by hybridoma, but in recent 4 years, around like 20% of the FDA-approved antibody drugs were discovered via the single B cell platform. Yeah. Comparing with other conventional antibody discovery form like hybridoma or phage display, single B screening is fast and also offer like a good diversity, as you can see from the case studies I'm going to show at the end of the talk. So apparently, Beacon, Berkeley Beacon, is the state-of-the-art and best in industry technology, for a single B cell screening. Yeah, as you can see, in terms of throughput, accuracy, automation, sensitivity, and multiplexing, Beacon based on optimal microfluid chamber is the best. Yeah, and also mentioned did some very good, like this introduction of how advanced the Beacon technology is nowadays. Yeah, and but similar to some other single B cell screening technologies, there are also some challenges for the Beacon platform. So first is that it's kind of pretty difficult to do functional assays for on-chip Beacon screening. Yeah, for example, a very powerful RGA assays cannot be done in the Beacon on-chip screening. Yeah, and also because the B cell on the Beacon chip is some kind of under stress, so its viability might be compromised after multiple rounds of screening for on-chip screening. Yeah, so usually you cannot do like over 3 rounds of screening. Yeah, otherwise the viability of the B cell might be compromised, and the successful rate in sequencing might decrease. Yeah, and also because the antibody concentration secreted by the single B cell may fluctuate, so sometimes the assay sensitivity of on-chip screening might not be sufficient, and sometimes you got false negative or false positive results. Yeah. And also because for Beacon, originally after you export the B cell, you have to sequence every B cell and do like plasmid preparation and recombinant expression for all the hits. So when the hit rate is low, it's relatively less cost effective. Yeah. So to address these challenges, the ProBio's team kind of upgrade the current protocol by combining linear expression cassette based technology, to this Beacon platform. So basically, we do a regular on-chip screening to identify the original hits, and then we export the B cell, and then we do a PCR-based, linear expression cassette, basically to amplify the antibody sequences, and then using transient transfection to do the recombinant expression, so that you do not need the very costly gene synthesis and plasmid preparation. And also the supernatant will allow you to do the high throughput functional screening based on the functional screening. And this is the comparison of the ProSpeed workflow versus the conventional plasmid workflow. Basically, after you export the B cell, you're going to do RT-PCR to amplify the cDNA, and using the cDNA, you construct linear expression cassette to do the recombinant expression of the antibody fragment. And using the antibody fragment secreted in the supernatant, you can do various functional assays, so that you can easily narrow down your desirable hits, from hundreds to like a dozen, so that you save a lot of cost and improve the successful rate to get functional binders. And I'm going to give you like 5 scenarios of this ProSpeed application. Yeah. So first, as I mentioned, using this ProSpeed expression, you can easily do functional screening of the antibodies secreted by the single B cell. So as usually, we do like a Beacon on-chip screening using the human cyno protein binding. We identify originally 116 hits, and then for these expert B cells, we construct linear expression cassette, and then do an off-chip, ELISA-based screening to only pick those top binders. Then we can narrow it down, to like 85, and then using this narrowed 85, the short list of antibody candidates, we do a RGA-based functional assay. So here is like IL-8 and CXCR1, signaling. By this RGA assay, you see the positive control inhibit almost 90% of the signaling, and some of the hits show like very potent inhibition of the signaling, suggesting they are likely functional binders. So then we successfully narrowed it down from 116 to only 16 functional binders. And these functional binders were also confirmed by the recombinant expression. So by doing this kind of process, we can successfully cut the original cost of around 90K to less than, to around like 40K. Yeah. A second application scenario is that, for the off-chip, screening, we can do multiple rounds of, or almost unlimited round of screening beyond the original on-chip screening limit. So in this case, originally on-chip, we did 3 rounds of screening. We test the human protein binding, cyno protein binding, and also the human cell binding. And we got originally 156, hits. And then we export B cell and prepare the linear expression cassette and use a high throughput transient transfection to get the antibody fragment secreted in the supernatant so that we can do off-chip screening. So here we are using the human and the cyno binding ELISA. We narrow it down, to 56 strong binders. And then we can further narrow it down by doing a fax to only, pursue those cell binders. And finally, we can do multiple round of counter screening to identify those, those binders without substantial cross-reactivity to the family members, so that we narrow it down to only like 14 specific fax binders rather than like original like 156. So by doing this, we can successfully cut the cost from over 100K to less than 50K. The third scenario I'm going to talk about is the improved assay sensitivity to reduce false negative. So sometimes, because of the high homology between family members, it's very difficult to identify, antibodies specific to one family member but not to others. So in this case, on-chip, screening, we do the human and the cyno, protein binding, and we do a counter screening against the family member F1. Yeah. Originally, we thought very few of the hits will cross-react with this undesirable family member. Yeah. But actually, after we do the off-chip, the LEC-based screening, we see a lot of those, binders actually cross-react with this F1 protein. Yeah. So by this improved sensitivity, we can successfully, narrow down the original 86 hits to only like 10 specific, hits without cross reactivity to the family member. Yeah, and again, it's a substantial reduction of cost from around 70K to around only like 20K. Another application scenario I'm going to talk about is that because of the improved sensitivity of the off-chip screening, we can be more effectively identify the true cell binders. For example, for some GPCR, like targets, because of the engineered cell express only very low level of the target on beacon screening. Sometimes because of the fluctuation of the antibody concentration, you can get some kind of false positive, signals showing that looks like you get many like cell binders, but actually for the off-chips, screening, because of the higher concentration, you only see like much less, true cell binders. So in this case, originally we exported 121 hits, but ultimately only confirmed like 24 of the hits are like true cell binders. And this is a summary of the highlights of the ProSpeed antibody discovery platform. So basically, this upgraded protocol is compatible with various functional assays, and it can do multiple rounds of screening beyond the current versions, the limits of the current version of Beacon, and with improved sensitivity. Yeah, and sometimes because of the nature of the linear expression cassette, you do nested PCR. Sometimes the improved sensitivity of PCR can also help you rescue some of the lost, RT-PCR, the hits. Yeah. Somehow that slide disappeared from this deck. Yeah. Okay. Anyway, so finally, I'm going to give you two like case studies. So one is the discovery of DLL3, antibody. So we started from immunization of two species of mice, and then we follow the conventional workflow of Beacon to do the on-chip screening, and then we use the ProSpeed expression to successfully narrow down the hits, and finally we do sequencing and recombinant expression and functional validation and characterization. So here you see. We immunized like 2 strains of the mice, and half of the mice were used for the Beacon ProSpeed protocol workflow. Another half is by conventional hybridoma approach, so that we can do a side-by-side comparison. Yeah, and this is the the QC, antigen QC and assay development, data. So basically, we do like off-beacon binding assays to QC the antigen, and also on-beacon binding assays to validate the reagents to make sure that everything is ready for the on-chip screening. And then we do like 4 rounds of on-chip screening. We do the binding to the human protein, cyno protein, and mouse protein, and also finally the cell-based assay to show to get the cell binders. And by doing this, we narrow it down. We totally exported like 2 rounds of like 77 clones, and also another 100 clone from the second round. Yeah, right. And then we do the linear expression cassette to do the functional characterization. Yeah, and here we see in a side-by-side comparison, the light bar is the LEC based binding assay. The dark bar is the ELISA using the purified antibody. You see a very good consistency between these 2 approaches. Yeah. And this is the sequencing result. Yeah. So this is we see some of the sequence in black color is from hybridoma. They tend to cluster into a certain phylogenetic tree. Yeah. But for the beacon, but the leads identified from the beacon approach, you see they kind of pretty much very evenly distributed to most of the branches, suggesting likely like a better sequence diversity. And what also we look at the affinity by ELISA and FACS and do a side-by-side comparison of beacon approach and hybridoma approach. So you can see from the beacon approach for this FACS binding assay, you get various hits with a spectrum of affinity. Yeah, but for the hybridoma, you get only pretty much like strong binders. You don't get too diversified affinity. And finally, in terms of epitope, we also did this epitope binning assay. So you see leads from the beacon, they show either compete with the benchmark or not compete with benchmark, suggesting diversified epitope. But for the hybridoma approach, pretty much you only get hits that not competing with the benchmark, suggesting that leads discovered from the beacon approach may have like a better epitope diversity. A recent, I'm going to show you another case. Basically, it's extended application of the beacon approach, so that we can do single domain discovery using the beacon beacon approach. Yeah. So here we immunize the alpaca, and then we isolate the PBMC, and then we do antigen specific memory B cell enrichment, and then we induce the plasma cell maturation so that this antigen specific B cells can secrete antibodies, and then we load the B cells, the plasma B cells, to the beacon, to beacon, and then do the single cell, screening, and finally we do, the recombinant expression and validation. Yeah. So by doing this, we successfully, and this is the immunization and good titer. Yeah. So by doing this, we successfully identify, 50 or 72, 75, positive sequences, using this beacon approach. Yeah. Right. And finally, you see the sequence diversity. This is very good. From the yellow is the antibody identified from the 3rd immunization, and the green is antibody identified from the 4th immunization. You see, we only got one identical unique sequence, suggesting that beacon-based single B cell screening is really can help us to get a very good diversity in terms of antibody sequence. So finally, I'm gonna maybe stop here. Yeah. So maybe running out of time. Apologize for the technical difficulty. Yeah. All right. Okay, I'm going to stop here and to see if there are any questions.
Okay, thanks very much for that. You could come back on screen. Right now your camera is off.
Okay.
But
turning on my camera.
Here we go.
Right.
we have the gang all here. Very good. Thank you both for the presentations. The audience is invited to add questions into the Q&A box. To start out with, I'll ask a question about whether memory B cells and plasma cells from the same animal can be isolated and screened using your system.
Yeah, so this was something that we recently kind of looked into, and we started to look into some of the mice that we were immunizing and harvesting spleen samples from. So we traditionally kind of harvest plasma cells directly from that, just so that we can kind of have a rapid workflow in a single day. But we started to wonder, hey, could we actually also harvest the memory B cells and start to activate those in a well plate as a second kind of set of diversity that we could get from that same immunized sample? And so we started to kind of do some paired experiments there. So from the same spleen, harvest both the memory B cells for activation in a well plate and screening in 4 days later, and then also the plasma B cells. We haven't done a full sequence kind of an analysis for that, just given that some of the antigens that we're using for immunization aren't so relevant or interesting, I think, to the community. But it is something that we've heard is particularly interesting. And we hope that as we continue to identify additional isolation markers for rat and even rabbit, that we can kind of start to really assess what the diversity looks like and how that looks against different immunization strategies.
Thanks very much for that. Now we have a question for Dr. Liang. Thank you for your presentation. Did the differences between the 3rd and 4th immunization occur in the same treatment groups?
So you mean immune response? Right. So for that case, I shared previously. Basically, we tried beacon screening using the PBMC from the 3rd and also the 4th immunization. We are concerned, that if we immunize too much, maybe some kind of immunodominant epitope will kind of skew the immune response to those specific epitopes, and then you are kind of lose diversity. Yeah, but in this case, fortunately, our data show that sequence diversity is still very good after the 4th immunization, because we only identify one identical sequence between the 3rd and the 4th immunization. Yeah, right. So we believe that because the nature of single cell based, the beacon is the nature of the beacon is single cell based. So you have like very good chance to identify antibody sequences with very good diversity. Yeah, right. It's not like some other like phage based panning. Sometimes you get whole plate of the same sequence. Okay, right.
And another question for you.
Right.
To establish your technology in the lab, do you need an extra machine?
Extra machine, you mean for? Of course, you need to have a beacon, right? Okay.
Yeah.
Okay.
That was an easy answer, then, wasn't it?
Right. Sure.
We may get a follow up on that, but let's go to the next question. Is there any risk of contamination between wells when performing on-chip mRNA isolation and cDNA synthesis? Yeah, I think I could take that. Yes, we have seen that. And certainly when we have lots of different secretors, because we're not necessarily fully capping those nanopen chambers today, there is a possibility, just given diffusion, for mRNA to be diffusing in and out of those pens, and there is some level of contamination. The hope is with a nice, viable, healthy sample that you're harvesting fresh, that the dominant kind of mRNA or BCR sequence that you're trying to capture more locally should dominate, and so we should be able to clean that out. But if
If there are really low viabilities or there's a lot of contamination or mRNA floating around, there is certainly a risk, and we have seen that. We are working to try to address that, but we don't currently have a solution out for that just yet. But continuing innovation, we are trying to address that.
And another question for Vincent. For you again, well, first, thank you for the overview. You provided, in which applications would you favor a cell over a bead-based assay?
Yeah, so in the slide that I kind of quickly went through about our cell-based binding assays, we really want to target this for antigens that can't be recombinantly expressed in a soluble format. So these are things like transmembrane proteins that don't fold correctly or don't create those the epitopes that or recreate those epitopes that we expect, especially when then translating those into recombinant antigens. And so we certainly would favor those cases. I will note, and Dr. Liang kind of covered this as well. Cell-based binding assays are a little bit more challenging to perform and to optimize for, and so there are some trade-offs there. But the nice thing about the system is that you're able to perform both if you have that opportunity, and so that is something that we typically recommend is to help funnel essentially and give you the most opportunities to find the right antibodies for any campaign.
Excellent. Thanks very much for that answer. I have one more question because we're almost running out of time, but this will give you an opportunity to let us know what new innovation or recent research leveraging the Beacon system are you most excited about?
Yeah, so this is, if you haven't caught some of the other webinars that I gave, I'm like going through a webinar spree right now. But we just had two recently, so you should check that out on our website. But we just recently got a chance to speak with one of our collaborators over at UCSF. This is kind of a really exciting area that I'm particularly passionate about, which is using the Beacon platform for autoantibody discovery. And so we were able to leverage patient samples, very similar to what we did for a lot of infectious disease work, but to discover actually a novel autoantibody that was responsible for neurodegenerative disease. And that was pretty cool. I think that's really exciting. It kind of showcases that the antibodies that we typically think about, I think, are really targeted towards therapies, but really can also unlock a lot of different areas and basic research for not just autoimmune disease, but vaccine development and epitope discovery even. So really excited about that publication that came out.
Well, thank you both very much. I think we've reached the end. Oh, did you want to weigh in on that question, Dr. Liang?
You mean the final question?
Yes.
Yeah, sure. Yeah. So currently, we're very excited that Beacon may also work for the single domain antibody discovery. Yeah. So I think this is a good chance to take advantage of the sequence diversity of Beacon, so that we can have more quality antibody, single domain antibody discovered, comparing with the conventional phage display approach. Yeah. So we are kind of doing more case studies to optimize some of the details, so that so far we have around 5 cases, but hopefully, or this kind of protocol can be one of the standard protocols for Beacon in the future, right? Right.
Thanks very much for adding that in. With no more questions, I will conclude this webinar by thanking Vincent Pai and Dr. Liang for their expert advice on how to accelerate therapeutic antibody discovery using innovative technologies to transform hit generation and lead characterization. And I thank you very much for joining the webinar today.
Right. Thanks, everyone.
An on-demand version will be available. I'll send that link to everybody. If you registered, you'll get the link. Please feel free to watch this and any of our on-demand webinars when it's convenient. Thanks again, and have a great rest of your day.
Okay, have a good one. Bye.